Hello, everyone. Thank you to those of you who showed up in person. Let me see if I can move the slides. Okay, there we go. I am personally very excited for tonight's lecture. I'm sure you are as well. I'm just going to say that on your behalf. We're going to be talking about applications of AI in omics. So all of those genomics, metabolomics, proteomics, and whatever one of those new things people come up with that has omics on it. You have a difficult job tonight. And oncology. And so Bahareh is going to tell us about this tonight. She is a graduate student in the Neuroscience and Mental Health Institute, and she does machine learning and genomics research. So without further ado, I will let her take this on.
Yeah, here should be. So can you hear me? Fine. Okay. Okay. Hello, everyone. Thank you, Esam, for the introduction. So today we're going to talk about AI in omics and oncology. So I want to start with this cute slide just to mention how we are actually going to talk about three very vast domains of fields of studies. Each of them can be approached from different perspectives. And what we are going to do today is just to talk about the overlap between them. And hopefully at the end of this session, you know about the oncology and omics and how can they work together. What can AI do for these both of these fields to work together, and what also the limitation that we have in applying AI in these two fields, and also talk a little bit about what is going on in research and also in clinic, and they are different, as you can guess. So we'll start by defining the oncology, and it's very general definition from a dictionary, which is it's a branch of medicine concerned with the prevention, diagnosis, treatment, and study of cancer. And on the right side, you can see the different subtypes of oncology in medicine. But what we are going to focus today is on the oncology research. And as you can see, actually, it is about cancer. And we know it is important because it is very common. And according to this poster from World Health Organization, 1 in 5 people will develop cancer before the age of 75. So before we move on, I have a question here from you, and I want to know, what do you think about this sentence? How informative this sentence, when you think about that, and you want to somehow map that on yourself or the people that you know? Do you have any idea? Zoom or in person? I don't see anything in chat. No idea. Okay, one message. And 100%. Yes. So yeah, it is informative in some way, because if I told you that it is 1 in, 2 million, then it was a different scenario. But at the same time, if you want to somehow think about all the differences that you have, for example, the place you live, your lifestyle, if it has already been running in your family or not, this number may change. And we will get back to this later, but that is one of the main important things that we are going to cover today. So we know that there has been already very advancement in cancer research, but still there are a lot of room for improvement. For example, we need earlier diagnosis, we need better treatment, we want to reduce the mortality. And part of the problem is because, and we know about also the problem and limitation, because cancer is a very, the complex cancers are complex disorders. If it is genes, if it is environment, also if their metastasis happen, and many people are also resistant to treatment. So to address all these problems, we can actually somehow summarize all this problem. And if we want to find the answer, we can sorry, it doesn't work. Okay, use this. Okay, then the answer may be through precision medicine, because as you can see the definition here, it is accurate medical intervention for the right patient at the right time. And it seems as if we have this approach, we may be able to address all those difficulties, limitations that we already we know about, we are aware of in oncology. And as you know about I don't want to go through the excuse me. I don't know why it doesn't work. Okay, it works now. Okay, it works. Thank you, sir. Yeah. So I don't want to go through the details of conventional medicine versus the precision medicine. I'm sure about you know already about that. But we know that when it comes to precision medicine, genomics and other omics are important part of that can give away some very specific and individualized information about the person. But it is not the only thing. And as you can see in this list, there are other things that should be also considered. And also besides that, we also know about when it comes to tumor, we have this tumor microenvironment, which can tell us a lot. And as you can see, this is a very busy slide, but that is how it is. I mean, we can talk about, these blue ones are tumor cell, and it is in this microenvironment, we have lots of other cells. And at the cellular level and at the molecular level, there are lots of information that can be used and already has been used in order to not only the diagnosis, but also prognosis and treatment. So these are also the information that can be omics information that can be used in studying cancer and oncology. So I include this slide for two reasons. So we can I can tell you about that is what is going on in real world. It is a process that should be, actually we are taking a step by step. And also it is somehow the outline of this talk. So we should start, if we wanna talk about precision and personalized medicine, which in this article use as the abbreviation is PPM. We should get the omics data, and then we need to process them. And then when we get to when we get to something, the algorithm that can be used, of course, it should be regulated. For example, as you know about, for example, FDA. And also, these are the things that we are going to cover today, but there's important part that we are not going to cover, but still we should acknowledge, and it is very important, and that is the economic and ethic aspects of this. We know that even if we have the best algorithm, it is not that all people can access them, and many things can affect that. But unfortunately, we get the chance to cover it, but it is important. So let's start by talking a little bit more about the omics part. So we know that there are different omics, as already Ehsan mentioned, and we can, to talk about this, we can go all the way back to the very basic molecular biology and central dogma that we have the genes, and genes are transcribed into RNA, and then RNA are translated into protein. But we also are aware of the epigenomics, which is somehow the effect of environment on the expression of the genes. So the information that we can get from sequence of the DNA is different from the information that epigenomic modification like methylation or acetylation can give us about the process of transcription and gene expression. But we also we know that it is, even we have more here. So even when we talk about genomics, it is not that we always, all the genes are, all the DNA sequence are going to transcribe or express in the cell. We have non-coding DNA, which are as important as genes and convey lots of information. Also, at transcriptome parts, we know that there are some RNA which are considered to be a structural RNA, and they are doing important jobs of controlling genes. And as you can see here, we are even more omics, like metabolomics. and also emerging omics, and they are already has been used in the field. It's not that we are waiting for them to be included, like radiomics, for example, microbiomics, which sometimes are categorized under genomics because they're actually the DNA extracted from our guts. And the things that I mentioned earlier, that even not, we can somehow get some information even about the environment or lifestyle from omics can be shown here, for example, because you know that about the microbiome and how your diet, how even your childhood experience, for example, if you play outside, if you have other siblings, can affect that. So I don't really want to confuse you here, but there are a lot of, it is a very expanded field of studies. And so when we talk about this omics, we should also talk about these techniques that enable us to get the data. There are a lot of them. We are not going to, of course, cover that part, but this is also gets back to that big circle that I show you, because there are a lot of things in this omics that all of them affect the application, because it is not only these techniques can give us the sequences that we are looking for, but even the pace or the expenses also somehow affecting the fact that we can use it in our research, because when we have more data, it should be, of course, it should be cheaper. So by this time, we imagine that we are, of course, many people work very hard in labs to get all those purified molecules that we are looking for, and it went through the sequencing, and we have the sequencing result. So at this level, it is supposed or expected for us to be happy and excited about the data that we have in hand. But most of the time at this level, we are facing a confused researcher or scientist. Do you have any idea why this is the case? No idea? Okay, sure. Okay, why do you think it's overwhelming? It's too much information. Yeah, and that is actually the case. So you know that about all this, there's some lack of resources, don't understand what the results mean. Yeah, they are all, yeah, it is confusing. It is too much information to deal with. So, and you always, you know, about this debate that we always face when we talk about machine learning, and there's always a question, what do you mean? You mean that people should be replaced by machine? But actually, we don't have this debate in this field, and it's a very peaceful field, because there's no way that human can go through the sequencing result. For example, in my own case, I have 23,000 samples and 300,000 SNPs. So if I want even to start with that, even just start a project, there's no way that I can find any pattern or compare them. So we somehow know that we need computational methods. And now the decision is to go with the statistical method or machine learning methods. And I know it is not even fair to put them so separate, because they have overlap. And in many times we use them both. But I put it in a separate actually direction to show that that is where actually the project starts. We should really know what is the aim of the project, what is the task, what we expect from the question that we are asking. And machine learning, of course, is not always the solution. And we are going to discuss about the question that can be asked and can be answered by machine learning. And I'm going to talk a little bit about that. I mean, what ML may offer in omics studies, and it can of course be applied in oncology, but at this level it's maybe more general. So the first and I believe most important part that I wanted to picture be somehow relevant to that is the prediction at the individual level, and it is the things that I started the talk with about that 1 in 5 person and developing cancer. When a physician is in his or her office and actually talking about the patient having cancer, the number that he or she is giving him about, for example, survival is informative to some sense. But when they want those numbers to make sense to that specific case, the numbers at populational level may not be that much helpful. So when we want to predict the prediction at the individual level, machine learning can help us. The second thing is about, sorry, I don't know why the numbers are all one. They're supposed to be 1, 2, 3. So of course, they should be in that order. So, and the second thing is about dealing with high dimensional and complex omics data. So it is actually the nature of the data, and we cannot do anything as we just mentioned. So it is the data, and machine learning can offer more techniques and tools in order to deal with this data. But of course, we should consider it's not that all machine learning can deal with that, but there are options for this. And the third one, which is the most controversial, I believe, one, is that for the prediction of using machine learning, we don't need the underlying mechanism. And I know that this is that can affect the application of that in the clinic, and we should actually use that with cautious. But that is one thing that machine learning can offer. So, for example, if I use the SNPs, and for example, to do some prediction, those SNPs can predict and do and fulfill that specific task, but they may or may not give away some information about underlying mechanism. So we don't need those mechanisms for the machine learning. That's why, for example, we use whole genome sequencing, while we are not aware of the role of all of those SNPs in the result. So with that, all those things as introduction, now we can talk about actually what is going on in research. And I wanted to mainly talk about the process that you see in the articles, or if you want to do to work in this field, step by step. And then I'm going to cover 3 real examples from the literature. So when we talk about application of AI in using omics in oncology, we are actually, at the end of the day, it's a machine learning problem or project. So we are following a machine learning pipeline and fellowship. So we start by posing the question. It is important. We already talked about that, and we are going to talk more about this. Then we should collect the relevant data, which is important. As you know, I'm sure you have heard about garbage in, garbage out. So it is important to have data. It's important to have enough data. And again, we talk about this specifically in oncology. And then the next step would be to convert the data into matrix of number. And somehow it may seem to be redundant, and that's the same. But I believe it is different because all these steps are heavily based on domain knowledge. So the people from oncology, the people from the basic research with all those, the experiences and the studies in the fields, they are aware of the characteristics of the data, the limitation of the data. And when you change them into numbers, computer doesn't understand them. So it's important for the people with the domain knowledge to try their best to convey all the information, all the limitation, to find the best model, or to convert the data in a way that can get the best result. So it is very important part. And when we get to this point, actually the miracle happens. So you change a oncology question into a machine learning question that can be understood by computer, that can be understood by people from with different other expertise like computer scientists. And then this data can be fed into the ML pipeline. And I know it is, I don't mean it should be always supervised learning, but I just mean that it can be any machine learning pipeline that we are aware of, we are working with. So, but what about this step? I'm going to talk a little bit about these 3 steps and then get into the different models that as different examples. So about the questions, it is actually this graph shows us different questions that already, actually they are, this is a review article, and it covered different questions that has been addressed in the oncology using AI, mainly deep learning. So it can be about diagnosis. So it can be to know about the cancer, automated detection of the cancer, or it can be about subtypes of a specific cancer. And it goes all the way to prognosis, for example, about survival prediction, and also the treatment, which is important. For example, if that person can of course help the doctor to choose if that person should go with drug A or drug B, which is important. And that is that the question can be in each and in one of these steps, and then we get to the data. So when we have the question, then it's time to get the data. And like any other project, there's two options. The data can be recruited. I mean, the group can recruit samples. It has its own benefits, as you can easily define what omics do you want, how it should be, if it is longitudinal or it's cross-sectional, so it is good. But at the same time, we know that time and expenses is something that most of the time limit the number of samples, so you end up having a smaller data set. So the second option, of course, is to use publicly available data sets. And I put it as another option, but I believe it is a very big deal. And also I can say the availability of these open source data sets in omics and oncology is actually fuel of all the studies that we've seen, all the AI and machine learning studies that we've seen in the field. Because this data, for example, this first one, which is, you see in most of the article using these techniques, have thousands of samples, and they are multiple different numbers of omics. So it enabled the people from even computer science to get those data and work on the challenges that we face, and we will very soon talk about them. Also, it is very good, for example, if we have some kind of very rare cancers. If you remember about the very beginning of the presentation, I brought those tumor microenvironments. So cancers for sure are different and unique, but if we go to the cellular or molecular level, they have a lot in common. So if this data is available, they can be used to study even when the data set is smaller or the cancer is rare. So it is actually a big deal that there are a lot of these open sources data that can be used in the field. And before I move on, I just want to say that even when we get the data, it can also affect the question. You may start by asking a question, then check the available data, maybe get back to the question and change it or fit it in a sense that you can use the data. But now that we talk about this open source this open source data that can be accessed, that actually somehow affect the rise in the using of multi-omics instead of omic in oncology. And as you can see here, this article is not that much recent, so we don't have all the data, but you can see the rise. And this is for the multi-omics and deep learning, and this is for machine learning. And as I mentioned at the beginning, of course, using multi-omics also has the other benefit, because we can get the comprehensive picture, because we can have all these things in, for example, genomics, then we can have the epigenomics, proteomics. Each of them can offer part of it, we can complete the whole puzzle to have a comprehensive picture. But of course, it's not always like any other thing in life. It's not always everything is good. It also may pose some kind of limitation. And can you think about any limitation or difficulties that using multi-omics may cause in chat or? No idea. Okay, I go ahead and yeah. So it is. So when we have more data, actually we are calling for trouble, because even if you have a very big data set, you're keeping the number of the samples constant and add to the number of features, add to the columns. So we ended up having high dimensional data, and our data is heterogeneous. So it can be really a big problem, because the things that we can think of, even the easiest things that come to mind is the storage or the calculation and process time. But it is just a start of the things that the complications. So as you can see here, there is even in this trend, you can see there are other fields, which is multi-omics and integration. And that is all the different methods that can be used in order to integrate data, how those data can be integrated, and still we have an efficient, we can run and develop an efficient model. So that is a big deal when it comes to multi-omics and AI in oncology or in any other medicine. But according to the data, according to the previous slide that I show you, because of the availability of all those open source, most of the multi-omics and AI research are in cancer and oncology studies. So now it is that we have the question, we get the data, we are aware of the limitation of the data. Now it is time to do the actually have the input and feed this data into machine learning. And as I mentioned, it can be like any other machine learning, with this difference that instead of having one data set, we have more if we work with multi-omics. So then the question is how we can integrate them. What are different options? What are different ways to integrate the data? So I just checked the chat. Okay, yeah. Okay, so we have different, so here in this article, they actually cover 3 different ways to integrate different data, and it's not very specific, it's general ways. And in each of these, I should mention that for each of them, we can use multiple different models, the machine learning algorithm, supervised and unsupervised. And I'm going to give you one example for each of these categories. In this example, as an example, they consider, for example, it's just imagine that we have genomic, proteomic, and metabolomic, and also phenotype data that you see the abbreviation here. So the first way to, so imagine you are working on a project and, and you have all this data. Of course, you're very happy that you can get we have a comprehensive picture. And now you want to see how you can actually integrate them. One way, as you can see, the empty place in this stage 2 of integration, you can just skip that and just merge all the data together. So you can see the data that you have at the beginning, you also integrate them all together and then feed into the machine learning. Of course, you have a high dimensional data set, you may apply different feature selection, but you don't go through any pre-processing and integration. An example of this is we go through one article. So the question, the task or the aim was to stratify patients into treatment groups based on predicted clinical response. That means they want to see if they respond to a specific drugs. To actually, I believe they check 15 different drugs or not. And as you can see, the data set comes from those open source data sets. And the input was gene expression, copy number variation, and mutational status. And what they want to predict is drug sensitivity. But they make it in a classification. So it was if they have low sensitivity or high sensitivity. And they just combine them and merge this data. And they use 3 different models, which are ElasticNet, random forest, support vector. And among them, as I highlighted that, they reported that random forest did a better job, and they came up with a precision greater than 0.8. And it was for 11 out of 12 drugs. And they wanted to, and you know that even the evaluation when it comes to into these types of studies is very important, because you want to see, for example, when we talk about precision, it means among all if they say that this person will respond to that drug, in 80% they are right. That is translated in that sense. But we can move over to the next, which is model-based integration method. And as you can see here, it is this difference with the previous one. And the data that we have here and the one that are finally integrate to one another into the model is different. They go through another steps. And here, actually, if I want to say generally what they want to do is to some extent reduce the dimensionality of the data. And you may think, okay, we were so happy and excited about getting or going through whole genome sequencing and getting all the data. And now we want to remove some of them. And that is why it is really important step. So actually, we are dealing with a study with two-step machine learning. So the first step would be to somehow find a way, which should be, of course, reliable way, to reduce the dimensionality. And then when we have that data, then we can merge them and go on with another machine learning algorithm to do the classification. So an example of this is this article, which the task and question was to predicting the effectiveness of drugs in cancer cell lines. And it is roughly like the previous one, but they mentioned that they not only use the molecularly targeted drugs, but also they use the conventional drugs, that they are not aware of the mechanism that I just mentioned about them. One of the things that we can use in machine learning. And of course, I also mentioned the data set that they use. Again, it is those open source data. And input was gene expression, copy number variation, and mutation data. And they want to know about the sensitivity toward that data. But as you can see, the difference between this one and the previous one, this another step, which is integration. And they use deep neural network autoencoder, as they mentioned it, as a feature learning in order to reduce the dimensionality of each of these data sets separately. And when they came up with this reduced dimension, they combine the data in this step, and they use elastic net regression on that data, and also report the evaluation, which is good. And they use sensitivity and specificity, which is among all the positive, how many of them are true positive or total positive, and also the same for the negative, which here it means if they respond to the drug or not. And finally, the third integration method, which is somehow like the previous one, but there is actually the difference is in just the machine learning part. Not the whole thing is the same. So as you can see, the data that they get and the data that they merge at to fit into the machine learning is different. And here they transform data. And the transformation is, they have different methods like graph-based method or kernel-based method in order to transform data. And somehow it means that they change the space of data to find how they can become, how the relationship between the data. So it's, as you can guess, it's unsupervised learning. So in this example, they have the task of predicting survival status of neuroblastoma patient. And they use microarray RNA sequencing and methylation, which is epigenetic modification. And they wanted to predict survival of neuroblastoma. And also, again, as you can see, they use open source data. And here for the integration, they use patient similarity network. And here I just want to show you generally that, what does that mean? So you can see when they have the data, they change it as the name implies, they transform it. And each of these nodes is actually a patient. And the distance between this patient is based on the feature that they consider. And then they have this for each and every of the omics. And then they should find ways to fuse them, data fusion. And then they can do that machine learning. Then they feed it into the machine learning. And in this study, they use different models and report the accuracy, which is 79% and 7% in two different data sets because they use one of them as validation. And they reported that deep neural network work better than the other. So these were some of the examples, but in these two slides, of course, I don't expect you to read them, but I just want to show you it is from a recent review article, which lists all the different studies that use multi-omics in oncology. And I just want to mention, because you already see some of those newer omics or data types. For example, this is cell-free DNA, which is about liquid biopsy that moving toward replacing biopsy with just getting the fluid like blood. Of course, there's a long way to do that, but this is a good example to highlight the importance of those techniques that we mentioned at the beginning. And also, I remember last week, we mentioned about, Michael mentioned about pathology images and how it is relevant to omics study, and they already use it in their studies. But up to here, we talk about the research and all these elaborated algorithms that can be used and good evaluations and performance. But all the things that we talk about and we mentioned is at research level. We should also think about what is going on in clinic. All those algorithms, all those models, do we use them in clinic or not? So for that, I used this article, which actually it was very interesting because it's a project funded by European Union, and the name is Personalized Medicine Trials, PERMIT. And actually they asked this group of scientists to go and search and see among all those studies, and it's not just about multi-omics, omics, and AI in oncology, which of them get into the clinic. And for that, they have these two, laboratory developed tests, LDT, or FDA approved or cleared. But all of them are FDA cleared at that step. And why just those limited projects get into that steps, and why the other one couldn't get to that step? And to just do this research and summarize the result. So among all the study, I just put them in these three next slides, just 9 among all of the things that has been done in the field. And as I mentioned, it's not just multi-omics, it's also omics in AI and oncology. Only 9 of them get to the LDT or FDA clear steps. I put these two at the first slide because this foundation one is the only one that use multi-omics, both the RNA and DNA, and the rest just use one kind of omics. So it is, for example, this one, which is, I believe, one of the newest one, most recent one, which focus also on the timing, and do they want to do the DNA to report approach? They use next generation for the personalized treatment options. And the rest, as you can see in these two slides, I just want to go through them fast. They all use DNA microarray genes. So we just talk about how excited we are to use all those multi-omics, but what we see in clinic actually is using a very simpler model, using very limited number of genes. Of course, they started by more genes, but they ended up having, and that is what we expect, because it is faster, it is cheaper. They all use just microarray in different, they started using mainly focusing on breast cancer, but now we have more that use in also prostate cancer, and this one, which distinguish between 50 different tumors. And this is all that we have in clinic. In the next slide, actually, we will summarize what is the difference, what makes them to this level, and why the other studies couldn't get to this level, at least by now. But do you have any idea? Do you have any thought on this? Why? What is the difference? What makes them being approved? No one. No idea. That's good. I finished faster because I put some time for discussion. Commonality of diseases, sample size, yes. Of course, sample size, yeah. And also commonality of disease. Methodology, success of conventional treatment. Yes, exactly. Methodology is an important part. Okay, I go ahead and show. So as you can see, what they have in common, they just summarize all their finding in this graph, which is very easy to follow. And it's start by clarity of scope and goals. Of course, that is what we talk about. A study documentation, model interpretability, integration of prior knowledge, a study design and sample size, and a statistical evaluation. And that is, they have different graphs to say which of them have cross-validation, which of them have the validation sets, and all the things that we are aware in the very standard machine learning or statistical methods. But as you can see, these two are the two things that are somehow, we talk about them, and these are the things that it is somehow hard to get when we use machine learning, because many of the models, many of the algorithms that we are very excited to use, is hard to interpret. And even putting that aside, sometimes even when we use the algorithm or models that can be, somehow we can get some interpret in some ways, but from the computer science perspective, sometimes it is not the right thing to do so, because we should just say, okay, we have this data and we ask the model to predict, and it is hard to get more information from them. Also, integration of prior knowledge is things that we also use in machine learning. But as I mentioned, sometimes we just use the data without any prior knowledge because we don't have that knowledge. But it seems as if it is one important factor that can help them get into the clinical use. So of course, it is roughly my last slide, and it is somehow ending with an open question, but I believe it is not that this question cannot be answered in this session. It is a question that we should think about, and it is to find a balance between all the things that we have in research and of course all the development and improvement, and what we really have in real world and what we need that. So to conclude and to summarize, we know that developing personalized medicine in oncology is the way to overcome existing challenges in oncology. Applying AI and omics data in oncology is the way to move forward toward personalized medicine. It can help us. And we talk also about the importance of all those open source data sets. We have plenty of innovative algorithms developed by computer scientists and researchers, but at the same time, we have very limited omics-derived tests available in clinical use. With that, I want to thank you all for your attention, and also I want to thank Ehsan because he went through my slides many times to tailor that for time and the audience. Thank you so much, and I'm happy to take any question if there is any.